Yes, thank you. Yeah, I'm a PhD student at the University of Basel, and this is a joint work with Valentin and my advisor, Ivan Duckman. As Emmanuel said, I'm going to talk how implicit reconstructions can naturally incorporate deformations, which the previous speaker talked about, and also in the keynote lecture as well. But it's mainly applied to the CryoET problem. Yeah, so I think many of you in the audience know what is cryo-EM. So cryo-ET is an equally important problem, but it's on a much larger scale. So here, yeah, so here is an example of the cryo-EM projection. So it's coronavirus cells, and they typically in cryo-EM, what people do is they look at smaller structures and try to reconstruct them in a much more detailed way. So the idea is you take these smaller structures which are repeated throughout the projection and stack them and perform cryo-EM reconstruction. But here we'll try to look at the reconstruction in the whole. So you try to recover the entire 3D structure of the volume itself. And now biologists mainly look at how these small structures, basically the spike proteins, are distributed in this larger cell images. Yeah, so to put it simply, what CryoET does is it takes the specimen, for example, a tissue cell, and freezes them, and now tilts and shoots electron at different directions. So the idea is that we get the 3D structure of the molecules in their native environment. So here's an example of a human cell nucleus, where you could see the distribution of various proteins on the nucleus boundary, outside and inside of the nucleus boundary. And the goal for biologists is to see how these proteins are expressed in a real environment, rather than doing certain chemical experiments and see the concentrations. So let's look at the forward model of the cryo-ET problem. So you take the structure, you shoot electrons in one direction and get the projection at the other side. And now you tilt the structure in certain known angle and repeat the process, and you get various set of projections. And typically, based on the microscope, these tilts are constrained. So for up to minus 60 to 60 degrees, and the projection should look something like this. You can see that there's a clean movement from one projection to the other. But this is a very ideal setup. So in practice, there are various artifacts which creeps into the data acquisition process. So typically, as people mentioned in the previous presentation as well, you get various mechanical shifts and rotations. This would be in the calibration process of people walking or talking when the experiment is going on, and also certain electron-induced motions. So we basically model these in the projections rather than looking at them in the volume itself. So let's look at some of these artifacts. So first being the shift. So you have the projection over here, which looks like this. And now the projection is slightly tilted, slightly shifted in one direction. You could see it shifted to the down direction. And we model this using a shift operator, which is defined by this parameter tau m. Similarly, for rotations, we use an in-plane rotation operator. So you could see that the volume is in-plane rotated. The idea is that the projections are rotated, so you don't need to consider other rotations when you're considering these deformations. And this is represented by the 2D rotation matrix, which is given here, and alpha m is the angle by which it rotates. And for other deformation, we mainly look at tiny shifts at different locations of the projection. So we model this using a time varying shift operator, which you can see here. Different arrow shows how these displacement fields of various points of the projection. And this can be represented by this function gamma m. So yeah, now let's look at how these projections are when we account for all these modeling. So first is the clean projection, what I'd shown you earlier. You could see that cleanly moving from one direction to the other. But when you see the real data, you could see that there's all these shaking artifacts which come into the picture. So our goal is to model these. But why do we need to do this? Let's look at what happens if we don't account for these deformations and how these reconstructions would look like. So typically in practice, people still use filterback projection, and it's stable, and it's, what's if there's a mistake in the forward model and how these filterback projection works. So, and mainly biologists still use filterback projection rather than other methods. So here's a slice of the reconstructed volume with the FBP, and we assume there's no deformation in this. You could see that large structures are recovered and also smaller structures are recovered. But of course, you have all these deformation artifacts. This comes from the limited angle of -60 to 60 degrees. But now, if there are deformations and you don't consider them, you get reconstructions which looks like this. You have all these ringing artifacts in the bigger structure, and also the small details are completely missing. Yeah. So let's. Yeah, and yeah, of course, there's a degradation in the reconstruction. Yeah. So to recap, the CryoET problem is we have this volume, we project it at different angles, typically up to 60 degrees with a difference of 2 degrees in them. And this can be modeled by this equation here given here. And P represents the X-ray transform. Yeah. And epsilon M is the noise. This comes mainly from the low electron beam intensity, what is used for the biological samples. And of course, now if you account for deformation, we have this function dm, which gives the 3 types of deformation, what I showed you before. And this involves local deformation, which I showed you, the shift varying function, and shifts, which I showed you in the first slide, and also in plane rotation operators. Yeah, so this accounts for our forward model. Now let's see how do we model the volume itself. In traditional sense, people used to use voxel grids or meshes to model these, model the volume, but inherently these volumes are continuous. So recently in the graphics community, people have been experimenting with implicit representations to represent the volume, and they've shown tremendous success in recovering many structures. So the idea here is they use a function to represent the volume intensity rather than a voxel grid. So typical architecture of this function would look something like this. You have a Fourier feature operator over here and use a large multi-layer perceptron to get the volume intensity at a given location XYZ. Even though this has shown great success, in practice, this takes a long training period. For example, to get a reasonably decent volumetric reconstruction, people wait for a day or two. And this is not exactly feasible if you want to speed up the reconstruction process. So recently, Mueller et al. Showed that you could have a specific architecture design for these functions, which significantly speeds up the reconstruction process. So the architecture design, what they use is multi-resolution grid and a small multi-layer perceptron. So the idea is, imagine you have a location XYZ and you want to know the volumetric intensity at this point. Use a trainable feature grid, which could be smaller than the volume voxel intensity itself, and you use some kind of interpolation. So typically bilinear interpolation is used to get the feature vector at this point. And you don't use single grid, you use multiple trainable grids of different resolutions, and you get different vectors at for the point, and you concatenate them, and you pass it through a tiny network or tiny multi-layer network, and you get the volumetric intensity. And they showed in practice that you could speed up the training process from days to a couple of minutes. You get a reasonable volumetric reconstructions. So we use this model for our representation of the volume. And now to parameterize the deformations. The trainable parameters are these MLP and the feature grid vectors. Yeah, to model deformations, we have 3 of them. So first being the shift, and the trainable parameter being here tau m, which is just a 2D vector. Similarly, for rotations, we just model the in-plane rotation using this the number alpha m. And for local deformation, which is slightly involved, I show you in the next slide. We have a trainable function gamma m. So including shifts and rotation, we have only 3 parameters to estimate per projections. Now let's look at the local deformations, the function which defines the local deformation. Yeah. So given an image, if you want to know how much it shifts from one direction to the other, first, what we do is we discretize them into a coarse grid, and we assume that we know the displacement vectors at these points. Now, to know a displacement vector at some point, we look at the neighbors and do bicubic interpolation to get the displacement vector at this point. So if we assume these functions, these parameters are to be estimated for NC number of points on the grid, we have 2 NC parameters to estimate. So here it would be 72 parameters to be estimated per deformation. Yeah. So now we have the model to we have the problem where we estimate the deformations and as well as the volume. So we jointly estimate both of them, learn both the volume model and as well as the deformation. For this, first we have the data fidelity loss, which is just the forward model, which incorporates the deformations and the projection. And second, we assume that these deformations are small. I don't think we can recover them if there are large deformations in the projection, and we use an L2 penalty on the deformations itself and jointly train them using the cost function given here. Yeah, so this gives us estimate for our volume as well as the deformation. So now let's look at some results of this model. So to qualitatively estimate the reconstruction, we use Fourier shell correlation. Basically, it does a cross correlation on the Fourier shells, on the shells of the volume, but in the Fourier space. And we compare it with the current state-of-the-art method called RTOMO. It's a software package which does alignment in rotation, shifts, and deformations. But they do it somewhat heuristically and also model-based. They look at if the projections are similar from one angle to the neighboring angle, and then they compare them. And yeah, and to compare our results with RTOMO, we use simulated data of SNR 10 dB. And of course, our volume size is of 512 cross 180. Typically, the volume what you get in real data is larger, around 3K cross 3K. I think we still are working on a higher volume etc. But our model does scale well to higher model, but we used these 512 cross 180 size models. So now let's look at the Fourier shell correlation of our method. Yeah. So here the x-axis shows us the frequency, that's the radius of the shell. This also gives us the resolution as well. As higher frequency component would mean more details are required, and this corresponds to the resolution of the volume. And you can see that since we do not use any kind of prior or regularizer in our reconstruction, we hope that FBP without any deformations in the volume is the best we could do. And of course, FBP standard is the one which uses projections with deformation, but it doesn't account for any deformation correction terms. And of course, and as you see, our method comes close or even beats at certain points for the in terms of FBP, in terms of reconstruction in the Fourier space. And now, typically, biologists do not like to study the volume, so they more rely on the FBP reconstructions itself. So what we do is, since our model has both the FBP both the volume reconstruction and the deformation estimation term, so what we do is we use the deformation terms, what we learned, and use the FBP to reconstruct the volume. And you see that, our term comes much closer to the FBP without deformations. And when we compare with RTOMO, it performs significantly worse, which we are still looking at. We are not exactly sure. Some hypothesis what we have is there are certain deformations, which I'll show you next. So next we look at the aligned projections. Yeah. So here's a clean projection, what I showed you earlier, like the movement between one projection to the other is much smoother. And when you observe the data, there are all these wriggling artifacts. But RTOMO does remove certain parts of the artifacts. But you could see that at the end here or here, there are certain deformations in the projections itself. We think that this accounts for the low resolution in the Fourier space. And when we look at our reconstructed volume, the projection of our reconstructed volume, it's much more smoother, and there are no deformations similar to what we observe in our tomo. So from projections, now let's look at the reconstructed volume itself. So here is, as again, as shown before, I have a slice of the volume reconstruction, and this is for clean, and this is for FBP without any deformations. And our tomo performs, gets at least large details correctly, but the smaller ones are still missing. And now when we look at our reconstruction, you could see that the smaller details are clearly present. And now let's look at the setup where we use our deformation estimated parameters, but we don't use the volume function itself. And yeah. So this is from the previous slide, the FBP, our tomo, and our method reconstruction. And when we just use our deformation parameters and run FBP on it, we see that the details are present and it's almost close to FBP without deformations. Yeah. And next, we look at the results where we compare our error with the true deformation errors, true deformation in the projection. And for shifts and in plane, since these are smaller parameters, our method accurately recovers them. But for local deformation, there's a bit of struggle, but it's still better than what our tomo does. One of the assumptions is that there are empty spaces in the projection where any deformations are true. So this is not accounted for in our model. Yeah. So in conclusion, we jointly model deformations and the volume estimation parameter. We learn the deformation parameters through the forward model rather than certain heuristics. And also our formulation can easily incorporate bias, which we do not do now. And it's computationally efficient. You could use certain tricks to make it much more computationally efficient and independent of the size of the volume itself. However, we do not look at large volumetric deformations, which are typically present in CryoET. Although current methods do not look at these deformations. And also, we do not look at the effect of limited viewing angles. Yeah. Thank you.
Thank you, Vinith. Do we have any question in the room?
I have a quick question. So when you represent the volume with an implicit representation, is how do you implement the forward model? So when you implement the projection forward model, do you still create a discrete volume, or did you find a way to implement that integration in a way that you can avoid formation of voxels in your forward model?
So we take a single ray and we sample points along this ray. And I think this would, it's not discretization because we randomly sample points along this ray. So it's a kind of a continuous, we don't voxelize the volume now.
Okay, but then in that case, because if you think of traditional implementation of filter, it's highly optimized, right?
Yeah.
And then if you do it manually now, because you need to compute this thing, do you lose significant compute time over it?
So I would say it's much faster than the more compute. The compute term what we looked at was in terms of training, but we didn't implement like a more faster version for the forward operator itself. We were more looking at the training process where to speed up the training process rather than the forward operator. Sorry. I mean, the reconstruction, when you're using the traditional implicit representation, it's much slower compared to the architecture. I think the speed of what we get is through the architecture rather than the way we sample the voxels, the volume itself. But it's still in the continuous domain, but we just need to discretize the integral of the projection. And this we still do the discretization.
Any other question?
Sorry if it's a naive question for tomography, but do you consider kind of shifts in the imaging plane and then rotations, but do you consider shifts in the kind of on-axis plane as well?
Also, so
Focus issues.
Yeah. So shifts on the on-axis plane wouldn't change the projection itself, right? So we want to look at further in the shift small deformations in the volumetric plane, which we are not doing currently now.
Okay.
Other question? I have one question. You model the deformation at the level of the projection.
Yeah.
Would it make sense to have the same modeling but on the volume directly? I mean, why is it choice? I mean, of modeling like at the level of the projection.
So I think we followed what standard methods were, which looked at deformation at the projection. And since if we assume a rigid volume, we look at only deformations in the projection itself. But I think deformations in the volume would be much harder, but we have not done it in this work yet. Still.
Thanks. Other question, maybe online. I don't know if there is any question.
So how would this model stand up to compressing the viewing angles? So if you did like compressed tomography where you don't take a linear section, how would that stand up?
So I think, can you repeat that question?
Yeah, so if you can subsample the viewing angles that you're looking at, so rather than taking every 2 degrees or 5 degrees, you take a random set of acquisitions. Could this model be updated to include that implicitly?
Yeah, I think when you do subsampling of the viewing direction, I think then you need to incorporate certain priors, which we do not do it here, but the cost function, there's no problem in adding a prior to data vector. Yeah.
Next question.
Thank you. Would it be interesting to incorporate other constraints coming from the Fourier domain of the volume, playing with the Fourier slice theorem, as it is done, for instance, in a previous work of Amit Singer with Cryo-EM reconstruction with unknown orientations?
You mean like the common lines between the different angles, right? Yeah, but I think the problem there is, since we model the volume in the volume space, we have not figured out how to efficiently sample points in the Fourier domain for the to efficiently learn the reconstruction from the R forward model. Yeah, but it should help us, but we do not know how to do this sampling in the Fourier space from our model yet.
Okay. Thank you.
We have time for last question if there is one, otherwise we'll move to the next talk. So thank you again, Vinith, for the presentation. And